This week in Bioengineering 101 from the Odin, we learned how to use a scale for our labs. We also learned how to pipette using a mechanical pipette. And we digested a fascinating paper on inducing pluripotent stem cells. So if you want to see that breakdown and more, stick around. This week's lab was divided into two very simple experiments, both aimed at getting us familiarized with simple lab equipment, starting with a scale for weighing things. For that, we used a pocket scale with weigh trays and spoons, LB agar as the medium we were weighing out, a vessel, and sterilized nitrile gloves, since we're going to be using this to create petri dishes for other experiments. I started off by wiping down all working surfaces with isopropyl alcohol, wiping off my gloves, the tabletop, and all the equipment we would be using. Next, I powered on the scale, put the weigh tray on it, then hit the tare button to bring it to 0. Then I proceeded to pour out the LB agar medium, targeting 5.25 grams. Once I had the desired amount, I put the contents into the vessel, and then added 150 milliliters of water according to the instructions. The rest of the process for creating agar petri dishes will be covered in a future video. So if you haven't done so already, hit subscribe and the bell icon to be notified. The next portion was focused on using a mechanical pipette. Pipettes are the most accurate way for measuring small amounts of liquid. So using a pipette correctly is an important part of this Bioengineering 101 course. So for this portion, we used a mechanical pipette, a microcentrifuge, pipette tips size for this 1000 microliter pipette, this orange tray, and green water to test the pipette with. On the front of the pipette, you can see the measurement dial. That tells exactly how much liquid the pipette is measuring and dispensing. The top of the pipette, where my thumb is the plunger. And the black piece at the bottom of the pipette is the tip holder. And my index finger is in the index finger cradle for ergonomic handling. Here I'm getting a feel for the plunger. The plunger has 3 positions. The resting position, where I am applying no pressure. The desired measurement position, which you can feel by sensing a light stopping pressure. And then the fully discharged position, and that is where the thumb plunger bottoms out. Also, by twisting the thumb plunger, you can adjust the measurement dial. Here I'm changing it to 100 microliters. In order to retrieve a fresh pipette tip, I insert the tip holder into the open end of the tip and pull it out. I then placed a microcentrifuge into the tray, which will function as a vessel for me to dispense the liquid into. I put the pipette tip into the green fluid and then pressed the plunger down to the desired measurement position and released the plunger, drawing the fluid into the pipette tip. To dispense the fluid, I pressed the plunger to the fully discharged position. There was enough fluid to practice with, so I pulled 2 more samples. The instructions advise to tilt the fluid so you can better see what you're doing. For the second one, I pulled a larger sample. This time when I pulled the sample, I made sure that the pipette tip did not hit the bottom so as to prevent air bubbles and a bad sample pull. As you can see, there are no bubbles at the bottom of the pipette tip. I attempted to pull a third sample, but my target measurement was larger than the available fluid, so I ended up drawing air in, meaning that this is a bad or inconsistent sample. When I was done, I hit the eject button to dispense with the pipette tip. We also read this fascinating research paper by Takahashi and Yamanaka about how they were able to induce pluripotency using 4 factors. Since this paper is beyond my current technical understanding, I tapped into my brother who studied biology in university and even managed a lab that processes some of the highest volumes of DNA in the country. So let's see what kind of an endorsement we get from him.
Cool. Let's see here. Here they talk about how like it has important medical implications. You could take a regular cell from the patient's own body and generate stem cells from that, and that would circumvent a lot of the ethical controversies or concerns around that. Yep.
So without further ado, here's my summary of this breakthrough paper. Pluripotent stem cells are cells that can differentiate into a lot of different cells. This paper provides an account of the process that the scientists used to demonstrate that the 4 factors they list here, OCT3/4, SOX2, C mice, and KLF4, are the 4 essential factors for inducing pluripotency in somatic cells. Takahashi and Yamanaka designated these cells IPS, or induced pluripotent stem cells. And these cells exhibited the morphology and growth properties of embryonic stem cells. These IPS cells, or induced pluripotent stem cells, were so much like embryonic stem cells that when they injected them into a mouse's embryo, they contributed to the mouse's embryonic development. And this has potentially huge implications for medical practices because induced pluripotent stem cells could be a way of circumventing some of the ethical concerns of using embryonic stem cells. The basic hypothesis that these scientists approached this problem with was that there are a variety of factors or proteins that are used for maintaining pluripotency in cells. So maybe some of those would be used for the induction of pluripotency in somatic cells. They started with 24 proteins or factors, and they did an experiment to figure out which of those were critical. In this figure here, we see that of the 24 factors that they tested, they were able to identify 3, 4, 5, 11, 18, and 22 as potentially critical. The science of this paper goes into a lot of detail, and as someone that's new to biology and in particular bioengineering, I wasn't able to follow 100% of the time. But on a very high level, I found this paper very fascinating because it showed me that cells that make up life are bidirectional. It's not that we go from germ cells to a small embryo to highly differentiated cells that grow and age until we die, but we can actually, through bioengineering, take a differentiated cell and induce a state in it so that it can become something completely different. And that is something that I think is super empowering, and I'm excited to learn more about it.
Yeah, I think it's important because you can, like you said, bring normal tissue cells back to that pluripotent state, which could allow for you to use those. They could be used in, therapeutically. If you need to like regenerate a certain kind of tissue or help, you could culture some pluripotent cells, put them in the right environment, and then they'll react to that environment. You have an egg gets fertilized and then it replicates itself, and then it's a cluster of cells, and then somehow all those cells are like the same and pluripotent, so they can do what some of them are gonna, their offspring will become different specific tissue, but at that point they're all just the same. So then what environmental factors based off of where it is in that development, how do the eyeballs start to develop, the mouth? So it formed into like, I think it's 3 germ layers. Like it's basically exoderm, the mesoderm, and the endoderm. So basically we're all like animals are just like tubes, right?
Quick note on why I'm taking this course and recording these videos. I'm a curious person who's fascinated by life and has a deep respect for engineering. And when I read Codebreaker by Walter Isaacson, I became intrigued by what's possible in the world of genetics. Then I got to this chapter on Joe Zahner, and that led me down the rabbit hole of biohacking. That's how I discovered the Odin. When I heard the term biohacking, I used to think of red blocker glasses and blending butter in your coffee. This is not your grandma's biohacking. This is deeper and potentially far more empowering. Week one is admittedly basic. If you want to be a computer hacker, you have to know how to use a keyboard and click a mouse. Using the scale and pipette is me learning how to type on a keyboard and click a mouse. So even though right now I'm just learning how to type, there's far more that's in store.